Okay, so I just introduced the concept of clearance, and I gave you some examples. If you didn't understand those examples, don't worry too much. I sort of feel like clearance is kind of this backwards way of thinking about how the kidneys handle solutes, and it's kind of non-intuitive. Um, but uh, yeah, so if you didn't understand, no problem. You can just memorize this equation. Okay, so we can calculate clearance by dividing the excretion rate of the molecule that we're interested in by the plasma concentration of that molecule. Okay, so essentially, kind of conceptually, if you want to think about it, what you're doing is you are taking the number of molecules leaving the body per unit time, and you're dividing that by the plasma concentration to get the corresponding plasma volume, right? And so remember that the units that we end up with are volume per unit time for calculating clearance. So why the emphasis on clearance? Well, it turns out that clearance is a really handy way of telling how the kidneys are handling a solute, okay? And what's really great about it is that you can calculate it in a non-invasive fashion. So looking back at this equation here, you can see numerator is the excretion rate of X, which you can figure out using urine samples. And then the denominator is plasma concentration of X, X being the solute that you're interested in. And you can get that by taking a blood sample, okay? So you can calculate clearance using just urine and blood samples, and that tells you a little bit about what's going on in the kidneys, which is much better than like cutting a person open and trying to look at their kidneys and seeing what's happening in there, right? Okay, so now that we've gone over how to calculate clearance, let's do a practice problem. Okay, so let's see if you can use the information in this figure. So there's various information kind of scattered throughout this figure to calculate the clearance of inulin. Okay, and what we're going to do as we do this calculation is we are going to assume that each one of these dots is 1 milligram of inulin. Okay, so go ahead and hit pause and see if you can do that calculation, and we'll come back to it. Okay, so hopefully you were able to determine that inulin clearance is 100 milliliters per minute. Let's go over how we arrived at that number. Remember that clearance of inulin is going to be equal to the excretion rate of inulin, which is 4 milligrams per minute, right? So 4 inulin per minute down here, divided by the plasma concentration of inulin, which we know from here, it was 4 milligrams per 100 milliliters, okay? And that comes out to 100 milliliters per minute. Now, there is something important that I would like to point out to you here, which is that you might notice that the inulin clearance is equal to the GFR, right? So we have inulin clearance equal to 100 milliliters per minute. GFR is 100 milliliters per minute. That is no coincidence. It turns out that, well, basically this, okay? So if a substance is not secreted or reabsorbed as it is processed by the kidneys, then its clearance rate is going to be equal to the GFR, okay? So a way to think about this is that basically for such a substance that's not secreted nor reabsorbed, then the rate at which it's going to be cleared is simply the rate at which it enters the tubule, right? So we have like our 100 milliliters with our substance going in. That same volume is going to be absorbed back, but without that substance. So that's the rate that it's going to be cleared at, right? So here we had our 100 milliliters of 100 milliliters per minute filtering. All of that gets cleared, um, and all of that inulin gets excreted. Therefore, our inulin clearance of 100 milliliters per minute is going to be equal to the GFR. Okay. So now that we know that inulin clearance is equal to the GFR, that's a really handy thing to know, because what that means is that we can just measure inulin clearance and that will tell us what the GFR is. And GFR is a really useful quantity for measuring in the kidneys. It measures kidney function, basically the amount of um, the amount of filtration that you have happening in the kidneys is going to tell you about your kidney health. Okay. Okay, so now it is finally time to address this pressing question that you've probably been thinking about this whole time, which is, what is inulin? First of all, I just want to point out inulin is not to be confused with insulin. Those are two different things, okay? Inulin is a polysaccharide found in plant roots. This is its structure, but its structure doesn't really matter that much for us. Really, the main thing um, about it is that it doesn't get secreted or reabsorbed by the kidneys, so it's a handy tool for us for determining GFR, okay? Um, Inulin is a little bit problematic as a tool because you have to, it's not naturally occurring in the body. So what you have to do is you have to inject it into the bloodstream, and then you can measure its clearance from there. So that's a little bit inconvenient because you have to hook a person up to an IV to get that inulin in their bloodstream. It's kind of invasive. So usually inulin is only used in laboratory type settings for determining GFR. Um, Clinically, we tend to use a substitute for inulin. We use this molecule called creatinine. So creatinine is a breakdown product of muscle metabolism, and it has a stable plasma concentration. And it's, for the most part, not secreted or reabsorbed. It actually does get secreted a small amount, but it's small enough that it can be used to fairly accurately estimate GFR in the body. So creatinine clearance is roughly equal to GFR. So this is a lot easier to deal with because you don't have to inject that into the body. It's just like a naturally occurring thing. Okay.